In Chapter 3, Segment 2, neurophysiologist Alan Hodgkin and Andrew Huxley studied a neuron involved in the emergency escape behavior of a squid. The axon was more than a half a millimeter in diameter, which is fairly big compared to normal mammalian axons, which range from 0.1 to 20 microns in diameter. They were able to see this axon with the human eye. They inserted microelectrodes into the axon without altering the properties of the axon. They even said that they can push out the intracellular fluid out of the squid axon and replace it with other fluid to see how the action potential is affected. They established that action potential is the movement of sodium ions into the cell through channels opening up in the membrane. At the peak of action potential, equilibrium potential of Na+ approaches about 40 MV. The concentration gradient pushing Na+ ions is perfectly balanced because of the positive charge pushing them out. The action potential then changes from a potassium-dependent resting state to the sodium-dependent active state before returning back to a resting state. This is completed through the actions of the voltage-gated Na+ channel. Voltage-gated Na+ channels are an Na+ selective channel that opens or closes in response to changes in the voltage of the local membrane potential. It includes a tubular membrane spanning a protein, but the Na+ selective pore is closed. The channel's shape changes when the cell membrane becomes depolarized to threshold levels. This allows the pore to open and let Na+ ions through. Na+ channels remain closed as long as depolarization is below the threshold. When depolarization reaches the threshold, Na+ channels open at first, allowing ions to enter the neuron. The neuron depolarizes the membrane and opens more Na+ channels. This accelerates the process until the barriers are removed and Na+ ions rush through because of the attraction to the interior, which is negatively charged. Voltage-gated Na+ channels, they open for a millisecond, then close again. Positive charge inside the nerve cells pushes K+ ions out of the channels that are constantly open, plus the voltage-gated K+ channels open so the membrane is even more permeable to K+. The resting state is now restored. When strong stimuli are applied, the upper limit of action potentials becomes apparent at about 1200 spikes per second. When more stimuli are applied, it showed that only the first stimulus is able to provoke an action potential. The axonal membrane is refractory or unresponsive to the second stimulus. Two refractory phases are the absolute refractory phase and the relative refractory phase. Absolute refractory phase is a brief period of complete insensitivity to stimuli. The relative refractory phase is a period of reduced sensitivity during which only strong stimulation produces an action potential. The voltage-gated Na+ channel monitors the axon's polarity and changes shape to open the channel before closing a millisecond later. This enforces action potential. The sodium-potassium pump enforces the low concentrations of Na+ ions inside the neuron. Axons carry out action potential. Meanwhile, cell bodies and dendrites have a few voltage-gated Na+ channels, but they do not conduct action potentials. In action potentials are actively propagated along the neuron, action potential near the cell body spreads a chain reaction down the axon length by speeds of 1 meter per second and in some 100 meters per second. Action potential is regenerated along the length of an axon. Conduction velocity is the speed at which an action potential is propagated along the length of an axon. It varies with the diameter of an axon, and larger axons allow the depolarization to spread faster. The myelin sheath that holds some axons is interrupted by the nodes of Ranvier. Nodes of Ranvier is a gap between successive segments of the myelin sheath where the axon membrane is exposed. It offers considerable resistance to the flow of ionic currents across the membrane, so action potentials jump from node to node, which is called saltatory conduction. Saltatory conduction in vertebrates gives them behavioral advantage over invertebrates. Invertebrates have small, unmyelinated axons, which makes conduction slower. They have giant axons that try and make up for this problem that immediate essential motor responses. An unmyelinated invertebrate axon would have to be 100 times larger to conduct action potential as well as a myelinated one. Scientists have found drugs and toxins that block specific channels, such as when a dentist works on a tooth, the drug stops action potential from sending signals to the brain. In synapses cause graded local changes in the postsynaptic membrane potential, action potential is propagated down the axon, and as the axon splits into many different branches, the action potential is recreated in each axon branch until it reaches the terminal of each branch. The axon terminal releases a neuron transmitter as the action potential's electrical signal is converted to a chemical signal. Neurotransmitters serve to communicate with the postsynaptic cell.